to our webinar series on avian influenza. My name is Rosita Dara. I'm an associate professor at the School of Computer Science, University of Guelph. I'm also the director of and founding director of AI for Food. I will talk about AI for Food later during my presentation. This webinar series is about avian influenza and has been organized in collaboration between Poultry Health Research Network and AI for Food. I'm delighted to open this important conversation together with my colleague, Dr. Buthu, to introduce a lineup of truly remarkable speakers, internationally renowned scientists who are at the forefront of their fields. Their work is shaping the global understanding of avian influenza and guiding the actions we take to manage its risk to animal, human, and ecosystem health. As many of you know, avian influenza is not just a regional or sector-specific issue. It's a global concern with significant implications for food security, animal health, public health, biodiversity, and international trade. In recent years, we have seen this virus behave in ways that have defied past patterns, crossing geographical boundaries, affecting new species, and creating complex challenges for disease management and policy. What makes this moment particularly striking is how much we know, or at the same time, how much we still don't fully understand. We have powerful genomic tools, surveillance network, and diagnostic technologies at our disposal. Yet questions remain about how this virus is evolving, how it moves through wild and domestic population, and how it may adapt in the future. That uncertainty underscores the importance of the work being discussed in this series. If we are to stay ahead of avian influenza, we need better predictive models, tools that can help us anticipate where, when, and how the virus might emerge, and informing strategies to mitigate risk before outbreak occurs. Developing those models depends on the deep, detailed understanding of the biology of the virus itself, and also environmental and other factors that impacts it, how it replicates, mutates, and interacts with different host species and the environment as well. Thank you for joining us today. I hope you find the presentation insightful, the discussions thought-provoking, and that together we can contribute to advancing knowledge and strengthening our collective response to this evolving global threat. With this, I'm going to pass it on to my colleague, Dr. Buthu, and I'm going to turn my camera off and take care of the team, kind of people joining. Dr. Buthu, please take over.
Oh, you're muted.
So thank you, Dr. Dara. And a little bit about PHRN. So PHRN was established in 2012, and we're a network of poultry researchers and poultry health specialists who address a wide range of issues from basic biology to environmental concerns, poultry diseases, production, and welfare. So PHRN at the University of Guelph consists of researchers and experts from various academic institutions, Public Health Agency of Canada, Ontario Ministry of Agriculture and Agri-Food Canada, poultry industry organizations, Poultry Industry Council, Livestock Research Innovation Corporation, and the Canadian Poultry Research Council. As well, there are more than 100 highly qualified personnel working on poultry-related research projects at any given time under PHRN members' leadership. So these include master's students, PhD students, undergraduate students, research assistant, postdocs, and visiting scholars. So the University of Guelph has had a long-standing commitment to animal health with one of the largest group of poultry scientists and poultry experts in North America. This combined with our state-of-the-art facilities make Guelph the ideal hub for global poultry research. So here at PHRN, we integrate cutting-edge research with high-quality education for next generation of poultry specialists and researchers. We also provide effective knowledge transfer to our stakeholders, including industry, government, and consumers. So with that, I would like to introduce Dr. Karsten Kirby. So Dr. Kirsten Kirby is a veterinary epidemiologist specializing in integrative disease modeling, combining field and registry data and epidemiological modeling to understand and predict disease dynamics across species and settings. He leads international and national research projects, including Enigma and PAIR, providing evidence-based support for authorities in managing outbreaks of avian influenza. So for today, Dr. Kirby will be presenting his work, which is recent advances in avian influenza epidemiology in Denmark. So please go ahead.
Thank you very much, and thanks for the introduction. I will try to share my screen. And I'm very happy that you have mentioned that there's a lot of stuff we don't know, because that's also what I'm going to touch upon in my presentation. Do you see my presentation?
Yes, it's clear. Go ahead.
Great, thank you very much. Yes, so I would like to talk about the recent advances in avian influenza epidemiology in Denmark, and I'm doing this on behalf of the whole avian influenza epidemiology group, consisting of Jan van Leeuw, Sandra Carpeto, Annette Boklund, Helena Anne Jensen, Lina Juncker, and myself. And there is a QR code if you want to look us up, because we have a website in the picture here where we try to put any news about projects and publications and other relevant things that we do. This is our boss, head of section, Hans Hove, and we are situated in the beautiful old veterinary school in the middle of Copenhagen. Right. So the questions I would like to address in this presentation is what the challenges are that we have right now, how do we address it, and what results do we have so far. And a brief overview of my presentation is that I will touch upon the avian influenza situation in Denmark. I will not go too much into detail with avian influenza because I assume the audience know that. Then I will refer to some statistical studies that we have done, and then the Enigma project and the PAIR project. So briefly about the avian influenza situation. Here you can see the graphs, the latest graphs from the EFSA report, showing the outbreaks of avian influenza in wild birds above and domestic birds below in Europe. And as you can see in the figure below, we have clear seasonality, where we have outbreaks during the winter, and less during the summer, and sometimes even none. But this pattern has been interrupted in during 22 in the wild birds, where we see large outbreaks during the summer. The other thing we can see from these graphs is that we have a lot of high peaks in previous seasons, but in two past seasons we haven't seen the same high peaks. So we have a changing transmission pattern, and it seems to be changing every season. It makes it very interesting, but also a little bit difficult to work with. And just to set the scene as well, in 21-22 we had almost 50 million birds culled in the EU due to outbreaks of avian influenza. So it's quite an important disease in Europe. Okay, so in the 2020-21 we had 16 outbreaks in poultry farms in Denmark. As you can see on the left-hand side map, the different patterns of the red dots indicate the poultry farms farm types, and the greenish dots, they indicate positive findings in wild birds. So we had 16 outbreaks in 2020 and 21, and this is because, or at least due, partly because Denmark is in a hotspot. So on the right-hand side, you can see a map from the bird migration atlas on the barnacle goose migration, and you can basically see that Denmark, or the southern part of Denmark, is based right in the middle of the flyway between Netherlands and Siberia. I'm tempted to say a highway because there's so many geese flying over Denmark and landing, and presumably also transmitting avian influenza. In 21-22 season, we had a different season. As you can see, we had a lot of coastal outbreaks, and we had 11 outbreaks in poultry farms. And in 22-23, we had 9 outbreaks in poultry farms, and we also had a lot of outbreaks in the wild birds. And finally, in 23-24, we had 13 outbreaks in poultry farms. So even though Denmark is a small country, we still can have quite quite a few outbreaks. And we haven't seen the last this season, although it has been less than we anticipated. But just to dive into this, to what we can do, is that we started with some statistical studies to sort of map the situation. And the first study I would like to mention is the one of Lene Juncker, who did a risk factor study on wild birds. And she found out that distance to coast and wetlands is important. And if you're closer to coastal lines or wetlands, you have higher risk of finding a positive case of avian influenza. But she also found out that this passive surveillance system that we have in Denmark, where the public can just report dead birds for testing, it is biased by accessibility. So we have bias in the data, and we don't know how to take that into account because we don't know how to estimate it. But that's just something we have to deal with. The next statistical study I would like to present is the one of Yang Van Liu, who did a risk factor study on wild birds and poultry farms, and she found that anti-reformers duck species have higher risk of being positive of avian influenza, and also the coastline and wetlands are risk factors. So we are getting closer to something that we can use in the models. And the last risk factor study I would like to mention is the one that was published this year from Helene Ane Jensen, who did a matched case control study on farms, where she found that observations of duck or gull species near farms were a risk factor for testing positive for avian influenza within the farm. And also there were indications of lack of bird deterrence that could be a risk factor for being positive. So we have some indications of some patterns that we can use to model. And this leads us directly to the ENIGMA project. ENIGMA stands for Early Warning System for Avian Influenza. And this is part of our research-based contingency that we do, funded by the Danish Veterinary Food Administration and also co-funded by the European Union, where we work on 3 different levels. We work on the continental scale, we work on the country scale, and the within flock scale. And I would like to touch upon all of these scales, starting from the bottom, where Annette Boklund and myself, we have been working mostly with our within flock model to address some basic questions like, how does an avian influenza outbreak develop in a flock? Is there any variation? How long will an outbreak take, and how many samples should we take if we want to test? And what was the likely time of introduction given that a poultry farm was found positive? And the basic aim is to be prepared for within flock contingency questions that we get from the authorities around these outbreaks that we have. So we built a fairly simple SEIR model, a simulation model, where we used weighted sampling of parameter estimates from the literature. So we had the beta, the transmission rate, with latency, so the waiting time until an infected bird becomes infectious, and the duration of infection. And we explored the parameter space within these estimates that we could find. And then we compared outbreak trajectories by species and subtypes. And some of our results indicate that we actually have quite a lot of variation. So on these plots, you can see the trajectories of the simulated simulations from the model. On the x-axis, you have the simulated days, and on the y-axis, you have the number of birds affected, and the red lines indicate exposed, and the green indicate infected. And as the left-hand arrow shows, we can see variation in the onset of the epidemics. And on the right-hand side, you can also see that we have different epidemic sizes given the different parameters that we put into the model. And then we can explore this So, for instance, we looked at the low pathogenic avian influenza estimates for turkeys, and we saw quite a lot of variation. Then we looked at the low pathogenic one for chickens, and we saw less variation, although as the arrow on the bottom indicates that we have possibilities of actually very slow developing epidemics. On the right-hand side, you can see now that the turkeys with the highly pathogenic avian influenzas above, they develop quite fast outbreaks, and we see kind of the same pattern for chicken with the HPAI, although we can also see in some cases can be prolonged. So this was purely to explore what could actually be happening based on the numbers that we have in the literature. So we can conclude that every outbreak is not the same, and it depends on the bird species and virus type, but also likely subtype and strain, which is very difficult because we don't have so many parameter estimates, and that more research is needed. And this is very handy because we have a new PhD student, Sandra Carapeto, who just started her project last year, where she will do an experimental vaccination study together with Wageningen University, and she will look into different effects of the vaccination with a more complicated within flux simulation model that she's working on right now. Right, so the next scale I would like to touch upon is the country scale. So Yang Fan Liu has been looking into her during her PhD project here. She has been looking into the spatio-temporal risk patterns within Denmark, and she has addressed research questions like surveillance, when and where should we look for avian influenza, and where is the risk, also transmission pathways, which ones are likely possible, and what are the possible control options that we could implement. And the whole objective was to create this HPAIV risk map for Denmark using a spatio-temporal simulation model with indirect environmental transmission through the environment. And Yang Fan has been using 5 key species to model this environmental transmission. The species that were most commonly found in the positive findings in Denmark before she started her PhD project was barnacle goose, Hooper swan, mute swan, and grey-lag goose, but also mallards, which were not that frequent, but we took that into the model as well because it's well known that they're capable of transmitting avian influenza, and they're basically everywhere in Denmark. And Yang Fan did a huge job with combining citizen science data on the left-hand side here from BirdLife Denmark, where she took the phenology curves, the distribution over the season of the 5 different species, and combined that with the novena bird counts, the winter counts, as on the right-hand side, where we have the spatial distribution and the numbers of overwintering birds in Denmark, done by Aarhus University. And combining that, she made a then HPAI wild model capable of estimating the spatio-temporal risk of outbreaks in Denmark in 10 by 10 square kilometer cells. And as you can see, still, even though Denmark is not very large, we have quite some local differences in the estimated risk. Yang Fan modeled both local migration, but also international migration in the model. And the epidemiological season starts in October as a common accepted way of thinking of it. And the output of this model was basically to say where and when can we expect any outbreaks. Yes, so going back to our statistical studies before, where Lene found that the passive surveillance data were biased with regards to accessibility, for instance, then we can look at the species-specific predictions and from Yang-Fan's model and compare it what we found in real life. And these plots show the red dots show the number of positive findings of each species over time, and the black graphs and gray areas show the modeled results. And as you can see, there's a good overlap, but it's not complete. So there are some things that we still don't know about this disease, or we have poor coverage of the positive birds in Denmark. Good. So to conclude, from the Dan HPAI wild model, it can predict the observed temporal trends, although comparison with surveillance data can be used, but it may be biased. And more experimental data on the virulence are needed. And also that barnacled geese are critical for transmission because they have a high abundance and a high infectivity. And Yang-Fan also found out that carcass removal can potentially reduce transmission. Then the last scale, the continental scale, you will unfortunately have to wait with that because that is Lene Jonker's area, and she will present that in a later webinar on May 29th. So I will encourage you to sign up for that one as well. This is going to be a very interesting series of talks. So while we're waiting for that to happen, I will go on to the next project that we have in line, which is also the project where Yang-Fan is currently working on. It's a PAIR project funded by the European Union, where we are looking at both COVID and avian influenza modeling. So we are basically utilizing that these two guys had a good idea at some point, that they made this instrument to detect virus fast, cost-effective or cheap, and it was field deployable. So you could basically take it anywhere and test for virus very fast, which is very neat in the case of an outbreak. So the idea was to take this PAN-POC, pandemic point of care system, and match it with a pan-risk part, the pandemic risk evaluation. So we had two parts in the PAIR project, where we, in the pandemic risk evaluation, which is the part of the project that we're mostly working on, where we're building essentially different models for different layers. So we have the wildlife with transmission of avian influenza in the red layer above. Then in the green layer in the middle, we have domestic animals, where we also have transmission, and we have spillover from wildlife to domestic animals. And then again, we have spillover from the domestic animals to humans, where we also have transmission of both avian influenza and COVID. So that is going to be very interesting, and it started last year. And the whole idea is to address the spread of disease very early on in the pandemic. So right from pre-emergence, to discover that we have something going on before it actually becomes a problem, so the authorities can implement timely actions against the spread. So both PAN-POC and PAN-RISK will be contributing to the combined risk assessment tool, where we have field validation and testing in veterinary labs and human healthcare situations for the PAN-POC. We'll have prognostic tools and spillover model and spatiotemporal risk modeling in the PAN-RISK tool. And then it will be combined into a surveillance system together. We have a lot of partners in the projects. We have 20 partners from, mostly from Europe, but also from the US. So that's very nice to have such great capacities. And the aim is to combine this fast screening with the available data to enable quick response to new outbreaks, to have this tool for quick detection and identification of pathogens and risk evaluation of potential epidemics. And in the end, it will basically be a tool for decision makers to evaluate outbreaks and to direct more sampling. Right. So, getting closer to the end, my questions in the beginning was, what challenges do we have? We have a lot. How do we address it? Mostly by modeling. And what results do we have so far? We have some results, and we are still working on quite a lot of approaches to get more results. So now you know a bit about what we have in the pipeline. And then I also just want to highlight that we don't always only sit inside the office. We also make a great deal of getting out of the office to see how the real world looks like once in a while. So these are some pictures from our excursions. And then I would like to thank our funders and collaborators, and thank you for listening. Thanks.
Thank you, Dr. Kirkeby. That was a very informative talk. I think you highlighted two major points, which is point of care diagnostics. I think there's a lot of interest in point of care and rapid detection of various pathogens, including avian influenza. And also the modeling. I think there's a lot of interest in modeling how not only birds move around, but also how do pathogens get around from countries. With that, I would like to remind everyone that if you want to you have any questions for our speakers, you can input the questions into the chat, and I will share the questions. So if at this point anybody has any questions, please go ahead and place your questions there. So we have one question here from one of the students. Daria is asking about Sandra's PhD project. Is she studying the effect of vaccination on virus transmission? Also, what kind of vaccine is she using in her project?
I'm not sure it's actually settled at this point in time. I can see that Sandra is online. So Sandra, if you want to comment on that, otherwise we're actually working on defining that right now. So I'm not sure it's actually fully settled, but she will be studying this transmission in some way. It's
yes,
very costly.
I think you brought up one. Yep, go ahead.
I was just mentioning it's very costly to do these trials, so it all comes down to what can actually be done in one trial because it quickly escalates.
So yeah, transmission is the name of what you've identified, which is modeling how viruses are distributing in various regions of Europe, and even how birds are moving. So how do you discuss transmission into a laboratory experiment, or how do you translate it actively? So you said you go out in the field sometimes, but is there more interest to really trap birds? Are you doing it in that sense, trapping birds and counting them? Are you relying on other pieces of information from various authorities to look at bird movement per se, so really get a sense of how many birds, the specific migratory waterfowls that you identified, to get a sense of numbers of them specifically?
Yeah, so we have a really good collaboration. We don't have time to get out and count the birds and trap them because we're such a small group, but we have really good collaboration with the University in Aarhus and also the BirdLife Denmark people, we have so many bird watchers in Denmark, and they have actually very good ideas of when and where the birds are. So we rely on those data, and they are actually made publicly available. And we also had some really good help from our friends over there to aggregate the data in a specific way, so Yang Fan could utilize it in her model. So basically, we rely heavily on our network to get these data for us.
So I'll take one last question from one of the members here, Bridget, asking is, in your modeling, are you able to include data on numbers of dead wild birds together with the data on detection of HPI in wild birds?
Well, it's sort of two sides of this. same. I'm not sure if I understand the question right. So dead wild birds is separate from HPA of positive cases.
Yeah, so I think that she's asking is mainly how many basically birds that are actually detected as dead or dying are actually positive for avian influenza, and what is basically the ratio that you're detecting?
That is a brilliant question. Actually, we're trying to get access to these data, but they are, sadly enough, they're not digitalized. So we have to find them in another way. But yes, I agree, that could be very, very interesting. Although it's still biased by the people actually going out there and registering dead birds. There's a lot of biased sources in that, and Young Fan can make a whole presentation, I guess, on that.
Okay, that's really good. So there's more questions coming in. I just want to remind people that at the end of both talks, we will have a general discussion section for about 10 minutes where I will take more questions from the audience, and we will get a reflection by. Okay, so thank you, Dr. Kukuri, for your talk. So our next speaker is Dr. Rosita Dara. Dr. Rosita Dara is an associate professor at the School of Computer Science and the director of artificial intelligence for food. Dr. Dara has established an interdisciplinary research program that spans applied artificial intelligence and data and technology governance, with a particular focus on agri-food systems. She has also spearheaded several initiatives related to digital agri-food at both the global and national levels. So these initiatives include harmonizing platforms for food traceability and transparency, governing agri-food data and trustworthy artificial intelligence, and enhancing automation in agri-food practices. So her agri-food automation projects covers areas such as developing surveillance tools for avian influenza monitoring and prediction, early identification of mastitis in dairy cows, and mushroom harvesting. So with that, today, Dr. Dara will be talking about AI for AI, which is leveraging the power of artificial intelligence for the control of avian influenza. So with that, Dr. Dara, please go ahead.
Thank you very much, Dr. Butu. And Carson, thank you for your great presentation. I just wanted to say that you're a celebrity in Guelph. I noticed that some of my colleagues are tweeting your papers. So it was interesting to see. It was a few years ago and it was without coordination. So I think your work is quite impactful. So so with that, I want to. So I as Carson was speaking, I just wanted to to clarify how my work and his work could kind of complement each other. And how AI can help some of the activities that Carson was just mentioning. My name is Rosita Dara. I introduced myself in the beginning. Thank you very much for attending the first webinar in the webinar series. We appreciate your participation. So I'll try to make it as quick as possible. But my research program, I'm a computer scientist. I work on, my background is artificial intelligence. I have worked on governance of AI and data. And basically, I apply my research to agriculture and food sector. So basically, my lab takes an integrated approach, kind of an ecosystem approach to tackling some of the real world problems. So if we focus on, as we focus on applied AI, we think about the governance aspect as well. And this is how, and obviously it's a system approach. We want to build systems that are useful to the end users. So we integrate both in most of the applications that we work on in my lab. So quickly, as Carson was mentioned, real-time integrated surveillance tools are considered as crucial priority for pandemic prevention. And also, although there are many tools, as I mentioned in the beginning, in the toolbox of the animal health authorities, decision tools, when I say tools, because I will go over a system development, but each of those components can be used as a tool for by emergency responders to make more informed and timely decision. So I called it AI for AI. As you know, there are different type of AI, in terms of terminology. But just want to quickly talk about how we use AI to improve some of the decision-making processes for avian influenza outbreak prevention. So one is prediction of risk. I go over them quickly, but if you have more questions, I'll be happy to answer. With the help of integrated data, representative data, and artificial intelligence, we are hoping to have a bit better prediction of risk. Building early warning tools, if data is accessible in real time, we hope to build tools that can help with early identification of outbreak incident in a region. Obviously, we want to improve optimizing outbreak response strategies. This means that we need data about response strategies that we don't have, but in the future, as Carson mentioned, some of this data is not available, but if we have information about past strategies, then we can evaluate and improve, and identification of high-risk populations so that proactively emergency responders can take an action. But I want to focus this discussion, because I'm a computer scientist, on building decision support systems that we are currently working on with my team, and some of my team members are attending this webinar. So we take a system approach to building decision tools that are hopefully useful by the end users, emergency responders, poultry industry, who were decision makers generally. So decision support systems are tools that take data from different, eventually when we build them, data from different sources, pre-process them, make them ready, and integrate them for near real-time or real-time decision-making. So we have lots of different sources, which I will show, and since the past 2 years, 3 years, data sources have become even more and more accessible, and they're high-quality data sources. Then so this decision support system has several modules. So we get the data, pre-process it, use machine learning or simple statistical tools, or just simply software code to clean them up. So that data becomes, we clean it up and put it under the data module. I will talk about it quickly. Then we have model module. It could be machine learning methods, or the statistical methods, or simple rule-based method that extract patterns and information from the data that we have collected, integrated data. Then we get that information, we extract rules from the data, and there are different ways of implementing decision support systems. Some of them have knowledge-based modules, some of them don't, but we can store those knowledge or rules into another module, and then we can get all of that information, visualize it for the end user, so that they can see data, they can see pattern, they can interact better with the system, and hopefully they find it useful. So as I mentioned, we have a data module. So these are some of the data sources that we are looking at. We have to keep in mind this data come in different formats. Some of them are very well structured. Some of them even have cleaned up. For example, CFI share avian influenza data with WHO. That could be considered very clean data, but we work with unstructured data as well, such as satellite images and search engine data. Search engine is okay, but social media data and many other data sources. So data come in different formats. So the first thing we need to do is clean and if needed, because some of the machine learning methods and artificial methods, they need annotated data sets. So we need to find ways, automated ways, manual ways to annotate data. Then we need to think about privacy and ethical aspects. So we proactively, in my research, we embed some of those features into these data modules. So we make it the anonymized data. The ID management would be a little bit more smart and strategic, rather than just dumping all the data into centralized format into the database, and then we integrate that information, that data, and we create linked databases. But all of this is iterative, so we don't do that without analyzing and knowing if the data is useful. So as I will show, so all of these steps, we build data module for a specific objective, for example, one that Carson mentioned, and then we go next step, integrate data, we build a model. Is it useful? We come back to the beginning. No, data wasn't pre-processed well. So this is kind of an iterative process that we go over. So I want to go over these sections very quickly. If needed, we will generate data. There are machine learning methods for simulation. We can generate data given the distribution of existing small set of data we have. We generate metadata, we create database schema. So that would be that module. I can give you an example of how it works for one of the objectives we are working. For example, my student Mina. So we thought of the idea of, so H5N5 has been reported on backyard farm. And in Canada, backyard farmers, they don't, poultry farms, they don't have to report their location, or they don't have to report that they exist, as far as we know. And we thought, okay, can we use satellite images so that we can extract that information in a kind of a, not in a privacy invasive way, but get an estimate of farm density, backyard farm poultry density, backyard poultry farm density, so that, this is an important indicator, an important risk factor. And can we get that information and have more accurate representation or value for farm density? Because some of that information is currently missing in Canada. So my student collected very small amounts of data, used best practices to increase the number of data, use very sophisticated, because this is unstructured data, and it immediately becomes extremely large. We use it very advanced ResNet and those kind of algorithms that are very deep and complex deep learning algorithms. And then we were able to, so far, we know our data is too small, it could be biased, because so we haven't still considered all other factors, but we were able to achieve 81% accuracy. Obviously, if we have more data, we can have a better estimation of farm density. So we still have to put a lot of effort on this, but this is just the data module. So we get images, we pre-process it, we extract meaningful indicators, values from the data, and store it in the data module. And then we have the decision support framework. It's something similar to what Carson presented, so I don't want to go too much in depth. So there is a decision criteria, there is a question. So we get data related to that question, pre-process it, as I mentioned before, build a machine learning method, statistical models on top of that, to be able to extract patterns. And if it is meaningful, great. If not, we go back to the data pre-processed. So all of the, as I mentioned, it is iterative. So until we get to a point that we trust the model, and then we can put another layer on it to make it explainable, and we move forward. So this would be the model module, and we store the algorithms and parameters that we have developed in that module. So another example, one of my students, Marzia, she extracted data from social media from different sources. From social media and also Google Google search scores. And then we analyzed that data with disease data. We looked for correlation, and we used predictive models to be able to see that whether we can predict avian influenza with social media information. Yes, we were able to show that, at least with the data in the past 2 years. We did a past study in 2017 on that as well. And we showed that we are able to predict avian influenza 1 week or even up to 3 weeks earlier in some cases. And so this was an interesting finding. Even visualizing social media data or Google data, it can be used as surveillance tool, even for countries that are not reporting avian influenza on a timely basis. Some activities in those regions can potentially indicate there is an outbreak in that area. So it could be a very powerful surveillance tool. So again, data collection, building data modules, having a question in mind, maybe for early identification of avian influenza, we were able to collect that information, all the relevant information, and make prediction. I don't want to go over sentiment analysis. We have done sentiment analysis and some interesting sentiment about avian influenza and concerns, or which I don't have to go over it. But then we have the actual decision support system. So which there was supposed to be an animation. This gray part shouldn't have been there. But anyway, so decision support system takes the decision support frameworks, models, and data that we have collected, puts those decision support framework, I mean, the questions and then models and analytics together to build a decision support system. So we take that, we design it in a way that data flow makes sense and all of that. And in the back end in the back of the system could be quite complex and sophisticated methods and models. But in the front end, we will try to make it as simple as possible. So rules could be extracted from complex data, such as the one that I showed you about backyard poultry and farm density or social media. But the front end for the end user would be quite simple. The indicators have been extracted, risk factors have been evaluated, values have been assigned, and rules have been extracted so that they can make more informed decision given the context of an outbreak. And so this is our goal. This is what we want to do. But it is a system approach. So basically, there's a system. Hopefully, in the near future, data could come in real time or near real time, gets processed, data becomes available, modeling happens, and then you have a user interface that you can see data in real time, and a dashboard that we want to build. But one important aspect of what we do is we want to engage the end users to make sure the tool we are building, we can do all the research without involvement of the decision makers and the end users. Basically, what we do would be pointless. So in our research, we try to engage as much as end users, and you can imagine how much it delays the research. But whether it's just decision makers, a CFI or poultry industry, or even at least researchers, epidemiologists, to engage them in different stages to talk about data, processing, maybe with poultry industry, about privacy concerns. So we want to bring all of that and make kind of, we call it privacy by design, ethics by design, or usefulness by design. We want to embed it into a system to make sure that what we are building can help decision makers make more informed decisions. They find it user-friendly, they find it trustworthy, they find it reliable. So as I mentioned, it is an iterative process. Lots of things that we talk with the end users. Some aspects are indicated here. But in the future, and I think one of our presenters may talk about this as well, we are collecting this massive amount of data in my lab, globally at Canada level. We are hoping to build a digital shadow when we can do it at a small scale. But then if we can get real-time farm data, we are hoping to turning it into a digital twin so that we can simulate and consider, kind of many different, kind of decision point, many different what-if scenarios in the future for avian influenza. But this is we are talking about 5 years from now, or hopefully shorter, but we will see. And here's some of the members in the team who have contributed to this research. This credit is not, I don't want all the credit. I'm a computer scientist. How can I get credit for this work? And thank you very much.
Okay. Thank you, Dr. Dara, for your presentation. That was a really, really good description of what is artificial intelligence and machine learning systems and how it can be used. I think so. We'll take general questions for Dr. Dara, and also we'll step into a general discussion here about the theme for today was really about artificial intelligence and machine learning building predictive models. And we saw Dr. Kirkby talking about predictive models and trying to describe and define where viruses and birds are going, and also how predictive models can be used to predict issues in agriculture, not just here in Canada, but around the world. So for both of you, you can chime in however you want. So in very basic layman's term, how would somebody get into AI and machine learning? Because there's a huge appetite to try to integrate general research with artificial intelligence and machine learning, and moving the research in a direction that maybe neither of us, anybody has ever predicted before. We've never thought about. Maybe machine learning can identify certain things, or artificial intelligence can give ideas about a direction to go into.
Yeah. So there is so if you know machine learning and you want to get into this subject, you have to educate yourself around disease modeling and a little bit basic, at least basic concepts of epidemiology. And if the other way around, if you know epidemiology, and that's great because you know statistical modeling, it won't be too hard. But then you have to educate yourself about machine learning. It is interdisciplinary, and that's challenging, especially for some of my students who come from computer science background. So basically, you have to learn the missing piece. So you have to take machine learning courses or introduction to artificial intelligence, or if you don't have any background, start with basic statistics and build on top of that one additional course around machine learning. Take your project in that area, and really machine learning is something you have to gain experience over time to get good at it, to become a real data scientist. So it's just practice, get involved in this type of projects that is related to your field or other fields, because we learn a lot, from other fields, kind of complex challenges, and you can come and apply it to the field of disease modeling. But basically, you have to learn what kind of skills you don't have and build those skills gradually. And obviously being involved in those projects could help a lot, because it may sound easy, but when you get more in depth into unstructured data, it could be, you need to practice, basically. I don't want to say it's challenging, but you need to practice.
Yeah, I can also chime in. So traditionally, we have been more old school to making more biologically founded models, where we sort of had a better understanding, or at least an idea of the individual parts. But recently, Jan Lene has been venturing into machine learning, and it really helps if you have a good idea of your system and how the parameters should look and the outcomes should look, because then you can easier detect if you're going down the wrong path. And it also helps hooking up with somebody who actually knows machine learning quite well. So that would be my suggestion to do.
Questions from the audiences for Dr. Dara. For your data, where were you getting your data? So I think you mentioned you were using the X platform and maybe Google platform also to extract your data to track mentions about avian influenza online?
For Google only, for social media? Or generally all data sources?
Generally, so it's just a question about how were you sourcing your data and which platform were you using in your studies?
Oh, lots of different sources. Carson can also talk about their own, their own region data. So we have some data from CFIA they have kindly shared with us, but citizen science data, it used to be for wild bird, but now I think our data is a little bit more structured now. Social media, obviously, you have to write an API, and then with with the current leadership of X or Twitter, if data is not accessible, it's not affordable, with Elon Musk changing it to be extremely expensive. Google has an API. Farm density is available. Satellite images, there are sources you can, you can register and get access to data, and with higher resolution, you have to pay. So lots of different sources. It goes quite a bit of an effort goes into collecting that data. So this is what when I teach AI courses, data machine learning courses, AI courses, I always say, by AI, I mean artificial intelligence, 91, sometimes 70% to 80 something percent of your effort is the data cleaning, integration. And then the 10% fun, 15% is machine learning and modeling. So it's not that much. It sounds fun, but it can be quite frustrating at times. But it is very rewarding at the end.
Yeah, I think that goes for basically any model that you want to build. You have to spend the time on the data first and doing the descriptive, and that it's really tedious. But yeah, other data sources we have been using, we have quite good registry data within the European Union that we can obtain if we just identify ourselves, and we have some quite good land coverage data, also citizen science data. Yeah, we use the WAR data, and Lena has also used the MPRESI data, but she will talk about that later. So different registries, open source data is basically what we used.
Yeah, so when you're building your models, either with machine learning, a specific algorithm, whether it's supervised or unsupervised, and how many times are you testing these algorithms? And what is the application in terms of adapting it to other fields? So let's say if your codes were open source for anybody of us, let's say for myself, I want to download and try to test it out on random data. Is it easily adaptable to other data sources, or how much effort would I need to go into trying to adapt your coding for my own purposes as well? I think because there's a general understanding of a lot of publications out there say data source are open source, but how easy is it for us to utilize it for other purposes as well?
So Carsten, do you want to go this time first, or do you want me to?
No, I will actually refer to Lena with that question because she has much more experience, but she's not here, unfortunately.
Yeah.
So if you-
It depends what other purposes that so with the decision support system, the way we are building it, and we have different modules. And for example, if somebody says, I want to learn something else from avian influenza, could you share, kind of your Twitter data? Yes, so we can share that information. And we have annotated, we spend weeks to annotate, a group of around few thousands of Twitter data, so we can share that information. It is useful in the context of avian influenza because the keywords that we have set in the API collected data about avian influenza, but I'm not sure if it would be useful in other contexts. It depends on really what your objective is. Getting access to that data, majority of them we don't use any confidential information, to be honest with you. At this point, we are hoping to get access to poultry farm data. They have sensors, bar condition, videos or images. We still don't have access to that information, unfortunately. So aside from that, most of the data we have is accessible. The data CFA has shared with us, that also, for the most part, exists with WHO. So there's WAHIS, there's a database. But you need to know how to write API to get access to some of that information, or you need to know a bit of coding for cleaning, because the purpose we use the data for may be slightly different from yours. So it could be that you get the data from the authors who have written the paper because the data is very clean and you want to do comparative study. Your objective is very similar to them. But if you have a totally different objective of data pre-processing, all the steps that I mentioned may have to be different. So it depends. So my answer is it depends on the objectives. And yeah. So I see lots of questions in the chat. So I'm wondering, I know it's 11 a.m. and I guess is it 5 p.m. or 6 p.m. your time, Carson?
It's fine.
Should we open 5? Okay, should we open it up to the audience? Maybe they want to turn on their camera to us, or should we read at least one or two of them? The remaining, maybe we can respond to it, or I can respond to them, and maybe Nitish, I'll send it to you if possible, and you can share with the attendees.
Yeah, so we can have a more general discussion now. So if anybody has any question, you can raise your hand and I can unmute you. Otherwise, you can always put your questions into the chat itself directly. So another interesting question is about data itself. So whether you're getting your data source from a local, let's say, Scandinavian area, or international from a global perspective, what is the importance of data privacy here and managing intellectual property? So because I think that's another big point of managing specifically source data. So what are your thoughts on it, and how do you go about approaching various institutions in terms of getting this data?
Thank you. So the way we approach privacy, security, governance, or ethics, we approach it from risk perspective. And by risk doesn't have to be somebody will hack the system and gets the data. I mean, that's obviously one aspect of it. So data has to be encrypted, and best practices, kind of access control mechanism. But it could be social risk, it could be ethical risk, it could be regulation. You have to comply. So having said that, so first of all, in Canada, and I think GDPR also, they don't protect farm data like personal data. So privacy is about personal data. Would farm data fall under personal data? Some aspects do, maybe location, but not a lot. So in terms of regulation, farm data is not that much protected. So it's not that we need consent for every single thing that we do. But we design our system in a privacy preserving manner. We proactively design in a privacy proactive manner. So for example, we anonymize, we de-identify as much as possible, generate synthetic data, instead of using real world data. So for privacy, so again, privacy doesn't apply to farm data as much, but if it does, but we proactively, at least my team, we try to protect it. Confidentiality and IP is different. Confidentiality, it's again, all the data that we have used so far, they were accessible. We have a research contract with CFIA. So those for confidentiality, you have to approach it from legal perspective, put an agreement in place, or if data comes with an agreement, read it and comply to it. So try to build processes in a way that you can protect data. So I saw a question on IP, intellectual property. So intellectual property, it doesn't apply to farm data, but if we use someone else's model, or if the data that they have integrated and processed, again, this is an agreement thing. So if I reach out to somebody who has collected that data, I have to speak with them to understand, what is the inclusion, what is considered, ethical issue or a boundary that I cannot pass, or how we can use that data. So all of that goes back to consent, research agreements, and that's how we approach it. I'm a researcher, so IP for me, I have never tried to protect what we generate that much. I mean, we have never done it. This is our role as a researcher, is to disseminate and share what we do. So that's how I have approached it in my lab.
Yeah, and also, so leading into that for you, Dr. Kukabi, which is when you're collecting the data, how do you decide which parameters you focus on when you're looking at transmission, either when you're correlating AIV in wild birds, and maybe even risk assessing its spillover into various mammals or humans? So are you looking at, like you suggested, the amount of virus in those birds, or the amount of birds that are coming up positive? Is it density close to a certain city center or something? Because you produced a map showing where these birds are positive and the regions around them. So if you tell us a little bit about your map and how you built it, and what is the risk assessment of that?
Yeah, so the map is basically, so the map is predicting detections of avian influenza, because we can only build models for what we have of findings, and we only have the findings that people report. We also have a little bit, and I didn't get to say that, we also have a little bit of active surveillance in wild birds, where we can direct some efforts into, for instance, game birds or other live birds, but it's not that much. So we heavily rely on the citizen science data. And even though we found that there was an effect of accessibility, or was a bias of accessibility, we do not take into account the distance to for instance, cities when estimating the risk to humans. We have basically avoided that in the past. But in this pair project, we will have a component where we'll estimate the spillover to humans, and that will involve both overlap in human density and positive birds on a larger scale, although than 10 by 10 kilometers. And it will also involve the genomic component. So what is the likelihood of spillover from the different strains that we see? And that is the plan for the pair project. But it's very difficult to estimate the spillover because it's a stochastic effect. We had, yeah. So it's really difficult to do.
So for both of you, do you think the models that you're building, what is the application in terms of the future, in terms of, can it be used to mitigate an outbreak? Can be used to predict the severity of an outbreak, or can it be applied in different regions? So not just Denmark, not just Canada, but other regions of the world if we have enough data. So if the data is not protected, like we're building on that argument of data privacy and data availability and so on, and collaboration with various, because I think we have many people from different parts of the world. So if they're ever interested in that kind of, what kind of data also are you looking for?
You want to go first? No. Okay, I have two major things here. So we have an expert group of avian influenza where we evaluate the risk of transmission within Denmark every week. And we use the model that Lene will describe later. We use that prediction model to estimate the risk, and it's actually made openly available, as we also talked about before. So we just try to provide the public with whatever we can do, and we made it openly available. And it actually estimates the risk to any country in Europe, or most countries in Europe. And what was the other one? I forgot. I was so enthusiastic.
Yeah, so can it be used to predict outbreak in various regions, or is it a model to estimate the severity of an outbreak? And also, what kind of data source are you looking for if you were to collaborate with international partners, let's say?
Any open source data that we can utilize. So right now we're looking into weather data and also to some extent genomic data to incorporate into the models, but it's really tricky because it's not so well covered, and it's sparse data. Even though it's big data, it's still sparse. So that's a tricky one to do.
Yeah. So few things I can address this question. First of all, I don't think the decision support system can be usable tomorrow, right? So because we are working on it, so much challenges that we have to overcome in terms of data quality, integration, even decision. Are these really helping decision makers? How reliable the decisions are? But different components of our effort will be useful. First of all, we are building models not only on Canadian data, but also global data. That will help our models to be able to generalize to different situations. So we are, at least that's our approach so far. So the model, how we have built it, what data we use, all these publications would be useful. So we are spending a significant amount of time to clean this data. So this data could be shared with other. So benchmark data and clean data sets are not, you cannot find them easily. So that's another way of collaboration. So we can share the data. The models that for example, generative models that can generate data that hasn't been studied with, which in the future would be useful with digital twinning, because you're building what if scenarios. So that also could be there are lots of interesting and useful outcome from this research. So it doesn't have to be a system approach. It could be different components of what we do. So that's how I look at it. In the future, would it be possible? Yes, if data from all aspects from modeling all the way to the decision and how the decision was implemented and what was the outcome. If we can build that system in the future, absolutely, these systems would be usable, and we can use it for surveillance in the future. But at this point, we are scratching the surface. I mean, there is so much that can be done. And also risk assessment. Backyard poultry is machine learning a good tool to calculate new farm density, to generate new farm density values, and would that be useful for decision making? You know, so all of those questions are still remaining.
Yeah, that's really good. I think that's a really good thought to try to conclude today's seminar, essentially a discussion, which is we need a lot of different source data, whether from large institution or small institution, and trying to also get people to be a bit more open about the type of activities that they're undertaking, whether it's small backyard operations or where are they even walking on trails, whether they are seeing dead birds or live birds, and the frequency of these birds presence in various regions. I think that will help a lot of not just park rangers, inspection agencies, tracking diseases and tracking bird movement in general. Yeah, I think machine learning and artificial intelligence is definitely the future. There's a big movement, especially in North America, pushing for studies and pushing for education in artificial intelligence. I think we will hear a lot more about it, not just in this series, but also in general, news and politics. And also from both of you, as well as we see more publications coming out. As we wrap up, if you have any final thoughts for our members today?
Not other than I'm looking forward to the next webinars.
Yeah, me too. Me as well. Thank you very much, Dr. Bhutto, for your great work moderating this this discussion. So I'm going to go over some of the questions and then maybe send a response maybe to you, Dr. Bhutto, and then you can share.
Sure.
But thank you very much for everybody to join us. It's an important dialogue discussion, and we look forward to the upcoming webinars.
Okay, so with that, I would just like to remind people that this is the first of several talks in this series that we'll be hosting. The next talk will be in a couple of weeks, so and I will be sharing links with individuals as we get closer to the talk. So if you have any questions or you want to reach out to our speakers today, please do let me know, and I can share your contact with our speakers if you're willing to, if you're interested in collaborating and working with them and learning more about their models that they're building and their work in general in their respective fields that they're doing. Even if you're a student, you want to get in touch with them, that's another great opportunity for you as well. Okay, so with that, thank you very much, Dr. Dara and Dr. Kirkaby, and also to everyone for attending today. I hope to see you in the next seminar series.
Okay, thank you very much.
We will.
Thank you very much. Thank you. Bye.
Thank you.